Assalamualaikum and a very good morning. A warm welcome to all participants from Malaysia, the SEAMEO Network and affiliated countries to IMR, to SEAMEO Webinar Series 2025. The theme today is From Discovery to Delivery, Translating TB Research into Impact. Our presenters today are from the Institute for Medical Research, Malaysia. I am Dr. Rozainanee Zain from Infectious Disease Research Center, IMR, and will be the moderator for today's session. For your information, the Infectious Disease Research Center deals with infectious diseases research, diagnostics, consultation, and training. The focus of research of this center is on leptospirosis, vector-borne diseases namely dengue, Zika, and malaria, antimicrobial resistance, influenza and other medically important viruses, tuberculosis, melioidosis, brucellosis, as well as other tropical diseases. There are 5 units under IDRC, which are bacteriology unit, virology unit, entomology unit, parasitology unit, and archaeology unit. We welcome any research collaboration under the scope of infectious diseases. Ladies and gentlemen, this webinar series was initiated with the aim in mind to share the knowledge, discuss topics on the biomedical research and present research outcomes. Before we begin with the presentations, allow me to announce a few housekeeping messages from the Secretariat. We will not be taking questions during the presentation, but participants may pose their questions using the link shared in the YouTube channel. The question and answer session will be conducted after both presentations have ended. An attendance link will be provided in the YouTube channel for those who would like to receive an e-certificate of attendance. Please subscribe to our National Institutes of Health Malaysia YouTube channel and click for notification whenever there is a new post. Ladies and gentlemen, for today's webinar we have 2 speakers. Our first speaker is Dr. Azura binti Mohd Noor. She is a senior research officer specializing in medical bacteriology at Bacteriology Unit, Infectious Disease Research Center, IMR. Dr. Azura began her academic journey at the University of Science Malaysia, where she obtained her Bachelor of Science with honors in microbiology in 2003. She furthered her expertise with a diploma in medical microbiology from SEAMEO Treatment in 2008. Her quest for knowledge led her to University Kebangsaan Malaysia, where she earned a master's degree in biomedical science in 2012. Dr. Azura then pursued a PhD in molecular biology and biotechnology, focusing on structural biology and crystallography at the University of Sheffield, United Kingdom, which she completed in 2019. She has a wide range of experience conducting biomedical research on infectious diseases at IMR from 2004 until now. She studies mainly focus on developing rapid and point-of-care tests, followed by drug development and vaccine discoveries. For today, Dr. Azura will be sharing her research experience with a presentation titled Tuberculosis, the Ancient Infectious Disease and the Challenges. Dr. Azura, the screen is yours.
Assalamualaikum warahmatullahi taala wabarakatuh. Thank you very much to Dr. Rozainanee for the kind of words introductions. Again, I'm Dr. Azura binti Mohd Noor from Bacteriology Unit, Infectious Disease Research Center, IMR. So my topic today will be tuberculosis, the ancient infectious disease and the challenges. Right, tuberculosis. Why it is called the ancient disease? TB pathogen was found by archaeologists in a mother and child in Athlete Yam, and now it is known as Mediterranean Sea, 9,000 years ago. The earliest written TB records were in India, 3,000 years ago, followed by China 1,000 years later. In the 17th century, TB was referred as the white plague due to the pale complexion of people with TB disease. And the first report of TB cases was in 1893 by the New York Department of Health and Hygiene. And tuberculosis is the old disease compared to the other infectious diseases. In 18th centuries, outbreak of TB disease sparked vampire panics throughout New England. And during that time, it's quite interesting. They call vampire panics because the person who died will come back to their family member as a vampire and start to infect other family members and cause death. It's quite interesting. But in March 24, 1882, Robert Koch presented his finding at the Berlin Physiological Society Conference on TB germ and proved that TB was an infectious disease caused by Bacilli Mycobacterium tuberculosis and not heredity. In 1905, Koch won Nobel Prize for this discovery. In 1908, Charles Mentu invented the skin test method by using a needle and syringe to inject the tuberculin. Followed by, in 1930s, American Florence Hebert, a PhD holder, developed a process to create a purified protein derivative, which is called PPD of tuberculin. And in 1940, the testing fluid, PPD, was adopted by the United States government. In 1921, vaccine. Albert Calmette and Jean-Marie Camille-Guerin developed the Bacilli Calmette-Guerin, which is so-called BCG vaccine, in 1921. In 1943, Salman Waksman developed the antibiotics streptomycin, the first drug to treat tuberculosis. Waksman received the Nobel Prize in Physiology and Medicine in 1952 for this discovery. And those figures are the most important people in tuberculosis history. Now we start with tuberculosis. WHO TB report 2024 reported that TB incidence in 2023, 2/3 of global TB cases were reported from 8 countries, led by India 26%, Indonesia 10%, China 6.8%, Philippines 6.8%, Pakistan 6.3%, Nigeria 4.6%, Bangladesh 3.5%, and Republic of Congo 3.1%. You can see in this globe the area of the TB cases, the TB cases from 8 countries, major contributor for TB cases. Incidence of TB cases 2023, a total of almost 11 million TB cases were reported by WHO in 2023, and 45% of the total TB cases are from Southeast Asia regions, and Indonesia is the second highest with TB case worldwide. More interesting, about 90% of infected individuals worldwide develop a latent TB and have a high chance of becoming an active TB. TB incidence. TB can affect anyone regardless of age or sex. And adult men is the predominant group of TB patients, which consists of 6 million patients. Adult women, 3.6 million, followed by children 14 years and below with 1.3 million. And the graph on the right side, you can see global estimates of TB incidence disaggregated by age group and sex. For MDR-TB cases, WHO report that the estimated number of MDR-TB in 2023 was 400 cases, and it involved rifampicin and isoniazid, the two first-line TB drugs. The net reduction in global number of deaths caused by TB. TB is the second leading cause of death from a single infectious agent after COVID-19, and it is caused by Mycobacterium tuberculosis. WHO reported that 1.3 million deaths occurred in 2023, where net reduction of death between 2015 to 2023 about 23%. And WHO actually target about 75% net reduction will be occurred in 2025. The agent of tuberculosis, Mycobacterium tuberculosis. Mycobacterium tuberculosis is a gram-positive acid bacilli, and it is resistant to gram-stain. It has no outer membrane and is non-motile. It is an obligate aerobe and does not form spores. It is a slow-growing organism and always appears in clumps. Its genome is about 4.5 MB in size and has high GC content, about more than 60%. And the genome contains many genes that encode enzymes for lipid metabolisms. And you can see here the figures showing MTB culture on selective media, Ogawa and LG media. And below are the microscopic figures of Mycobacterium tuberculosis under two different staining. One Zinn-Nielsen staining, and another one is Ramli staining. Its cell walls contain thick peptidoglycan layers, just like other gram-positive bacteria. The cell walls also contain long-chain fatty acid, which is mycolic acid, and lipoarabinomannan, which is arabinogalactan. And you can see the close-up image of Mycobacterium tuberculosis colony on solid media. It grows as a clump colonies. And below is transmission electron microscopic figure of Mycobacterium tuberculosis, showing that this pathogen is bacilli, root shape, and lipids contributing to waxy envelopes that make MTB hardy, impenetrable, and hydrophobic. The distinguishable properties of Mycobacterium tuberculosis, which is cell envelope. This cell envelope of Mycobacterium tuberculosis is very distinct from other bacterial cells. It has no outer membrane, very clear that on top of the capsule-like material, you can't see any outer membrane. And 60% of its mass is lipid layers. You can see very clear mycolic acids, and then below arabinogalactan. And then lipids avoid actually the pathogen from lysis activities of phagocytosis and are capable of intracellular growth, are resistant to drugs, and make them slow grower organisms. And below arabinogalactan, you can see very thick peptidoglycan, just like other gram-positive, followed by inner cell membrane and cytoplasm. Right, now we go to first-line drugs to treat MTB infection. If you can see here that about 2 or 3 of the first-line drugs targeting cell wall of Mycobacterium tuberculosis. except rifampicin. We start with isoniazide. The target site of isoniazide is mycolic acid layers, and the gene that responsible for it is CATG INHA, followed by ethambutol, where the target site is arabinogalactose layers, and the encoded gene responsible for that is EMBCAB, followed by pyrazinamide. The target site is inner membrane. We can still conclude inner membrane as one of the layer of cell envelope, and the encoded gene responsible for it is PNCA, and followed by rifampicin, where the target site is on DNA or RNA polymerase in the cytoplasm, and encoded gene is RPOB. Based on this figure, you can see that all the cell envelopes become the target of MTB drug. But what else left in this figure? It is peptidoglycan. And peptidoglycan here is one of the most important structure for the MTB cell envelope. Why? It is very important for MTB cell envelope. And probably peptidoglycan can be our next future promising MTB drug. And why peptidoglycan again? Because there are two things about peptidoglycan. Number one, its biological function is to retain or sustain the structure integrity of bacterial cell. If we interrupt any synthesis or inhibit the management of the peptidoglycan, we can directly kill the bacteria. And another one is peptidoglycan you can't see in human cells. And it is quite interesting if we can target peptidoglycan, because when we target peptidoglycan, we will directly target the MTB itself, not the human cells, so that the drug will be less toxicity to the human. Right. Brief introduction on peptidoglycan layers. At least we get some idea about peptidoglycan layers. Peptidoglycan consists of glycan strands comprising N-acetyl-glucosamine or NAG and N-acetyl-muramide residues that are cross-linked by short peptides beta 1 to 4 bonds. And peptidoglycans looks like actually mesh-like layers. And the chemical structure of glycan strand, you can see that it consists of alternate NAG and NAM. Challenges in TB management towards the goal of end TB strategy by 2030 and 2035. Number one, the appearance of multi-drug resistant MTB and the current TB HIV epidemic associated with its severe social implications, treating and preventing TB have represented a constant challenge in TB control. The second one, a large latent TB infection pool, which together with risk factors for active disease, global aging, slow and insufficient case detection, greatly slow down the decline of TB burden. And the last but not least, current pandemic and political conflicts. Moving to the next slide is more on research and research on progress, which we are doing now in IMR, consists of, number one, development of point-of-care test, POCT, for latent TB detection. Why POCT for latent TB detection is very important? Because identification and treatment of latent TB infection can substantially reduce the risk of development of disease and are important for TB control strategies. We do have already current tests to detect latent TB, but why we still need another POCT to detect latent TB in Malaysia, especially, and later in the world? Number one, tuberculin skin test or MEN2 test. This test has low sensitivity and specificity. Why? Because the antigen used in this test to trigger the interleukin or cytokines in human cells are not really specific to Mycobacterium tuberculosis. It consists of proteins that are come from not only from Mycobacterium tuberculosis, but also from other Mycobacterium species, especially environmental strains, including Mycobacterium avium, intracellular, fortitium, Gordonii, and so on and so forth. Next is interferon gamma release assay, which is IGRA. We do have two types of IGRA. We do have interferon gold, and then we do have TB spot ELISA. And then those IGRA use antigen containing ISAT6, CFP10, and T7.7. And then those have high sensitivity and specificity. However, those IGRAs have low predictive value. Why? Because the antigen used cannot be used to discriminate between active TB and latent TB. And both the ST and IGRAs have reduced sensitivity in immunocompromised patients. So what we have to do is, next, is developing POCT, new POCT for latent TB detection. What we are doing now for current research is investigating and identifying highly predictive MTB biomarkers to discriminate latent TB from active TB. And next, we are going to develop and optimize a new POCT for latent TB infections. Second research is development of a novel anti-TB drug targeting peptidoglycan of Mycobacterium tuberculosis. The current research that we are doing now is investigating and identifying protein markers, lysine homologs, responsible for peptidoglycan management in Mycobacterium cell walls. And then next, what we are going to do is constructing three-dimensional structure of the promising proteins using crystallography approach to elucidate chemical structure of the ligand peptide for drug development. So, first result that we do have is X-ray structure of Mycobacterium lysine, one of the homologs. And then this structure was solved and refined at 1.25 angstrom. Based on this structure, what we got, the information that we got from this structure is the proteins, the lysine, binds tightly to peptidoglycan analog. And then we can see on the structure itself, some of the conformational change happened within the amino acid residues. And the interaction between the peptidoglycan analog and the protein also can be further analyzed using this structure to get more information on how the interaction happened. To further confirm that, to further confirm how the peptidoglycan analog and the protein interact, to get some idea about the geometry of the peptidoglycan, the physiology, the binding pocket, all those interactions, the binding affinity between those letters was done by doing superposition of complex structure of Mycobacterium lysine peptidoglycan analogs with other X-ray structures of lysine protein from other bacterial species. And it's very interesting that based on the detailed analysis, we found that lysine, even from other species, not only from Mycobacterium, share a similar binding pattern. And actually, that is a very big breakthrough for us about how the interaction happened, the geometry, the chemistry, the chemical structure, and all those things that will be used to design our own MTB drug. And if you want to know more about that, I cannot reveal here, but you can email me further. And will be my last slide. Tuberculosis is the second leading cause of death from a single infectious agent after COVID-19, and it is caused by Mycobacterium tuberculosis. 45% of the total TB cases are from Southeast Asia regions, and Indonesia is the second highest with TB cases worldwide. About 90% of infected individuals develop a latent TB and have a high chance of becoming an active TB. The appearance of multi-drug resistant MTB and the current TB-HIV epidemic, associated with its severe social implications, treating and preventing TB, have represented a constant challenge in TB control. And the last but not least, WHO targets to achieve 75% net reduction of death in 2025. With that, I thank you.
Thank you, Dr. Azura, for the interesting talk. If there are any questions that you would like to ask about her presentation, kindly submit your questions using the link provided in the YouTube channel. Next, I would like to introduce our second speaker for today, Dr. Siti Roszilawati Ramli. Dr. Siti is a clinical microbiologist and a biomedical researcher from IMR. She is a medical doctor with a Master of Pathology in Medical Microbiology and obtained her PhD in Molecular Microbiology from Germany. Her niche areas are tuberculosis, zoonotic and tick-borne diseases. Currently, she is in charge of TB diagnosis and research at Bacteriology Unit IMR. It is our great privilege to have Dr. Siti with us today to share her research. Her presentation title is Decoding Drug-Resistant TB in Malaysia, a Genomic Perspective. Over to you, Dr. Siti. The screen is yours.
Thank you very much, Madam Chairperson. I'm Dr. Siti Roszilawati from Bacteriology Unit, IMR. So TB is a significant public health challenge. The emergence of drug-resistant TB has made the detection and management of TB become much more complicated. By understanding the genomic perspective, we can plan for better strategy to tackle this deadly problem. So in this presentation, I'll be covering the topic for tuberculosis burden in Malaysia, whole genome sequence in drug-resistant TB study, evaluation of targeted next-generation sequencing assay in drug-resistant TB, and last but not least, the discussion and conclusion. TB is a major cause of ill health, and it is the second world's leading cause of death after COVID-19. WHO has estimated that more than 10 million individuals have become ill with TB, and 1/10 of them died in 2022. Nearly 500,000 of new cases of rifampicin-resistant TB was reported in 2022. WHO has set targets to achieve end global TB on 2035 by reduction of TB death by 95% compared to baseline in 2015, and reduction of TB incidence rate of 90% compared to baseline. And the key components in Pillar 1 has called for early diagnosis of TB and universal drug susceptibility tests, highlighting the critical role of laboratories in rapidly and accurately detecting TB and drug-resistant TB. Mycobacterium TB complex constitute a genetically homogeneous group that is 99.9% similarity at the nucleotide level. The resistant mechanism in MTBC is not mediated by plasmid nor horizontal gene transfer like many other bacteria. So evolution of drug resistance mainly by chromosomal point mutations that lead to nonsense mutation as well as missense mutation. And these are most likely due to the selective pressure. By studying the mutation conferring drug resistance, we can highly rely on the mutations in detecting drug-resistant TB. WHO has recommended the use of NGS in TB, particularly for whole genome sequencing, in studying the transmission analysis, drug-resistant survey and routine surveillance, epidemiology, as well as evolution study. For targeted next-generation sequencing, WHO has suggested TNGS to be used for TB drug susceptibility tests to overcome the limitation of phenotypic testing that are long TAT, high-level biosafety infrastructure, poor reproducibility, and uncertainty for critical concentration for some drugs. And also the WHO recommended rapid diagnostic, which use limited resistant associated target regions. For Malaysia TB statistic, TB incidence rate was estimated to be around 122 incidents per 100,000 population in 2023. And this has made Malaysia as one of the high burden TB country, similar to its neighboring countries like Thailand, Indonesia and Philippines. TB death in 2023 is around 3,000 cases, with majority in HIV negative patients. Selangor and Sabah are the two main states with high TB burden. The TB incidence rate in 2023 has shown there is increase in 37% compared to the baseline in 2015, whereas TB death case in 2023 has increased to alarmingly 78%. The incidence of multi-drug resistant or rifampicin resistant TB in 2023 was estimated around 400 to 800 cases. However, this estimation was suspected to be under estimation. Whole genome sequence TB study was performed between 2017 to 2019 between IMR and IPR, and a paper on drug-resistant TB was published in 2022, investigating the lineage and resistant genotype pattern among rifampicin-resistant TB population in Klang Valley. The study site was done at the IPR, which is the reference center for TB cases resistant to first and second line anti-TB drugs. Around 56 clinical MTB isolates from sputum and pus were collected from known phenotypic rifampicin resistant and other drug resistant cases. And the sample or colonies from the MTB were extracted for genomic DNA and performed library preparation using Illumina Nextira library prep kit, and whole genome sequencing was performed using MiSeq and analyzed using online bioinformatic pipeline specific for TB genome. First finding shows that the main lineage for drug-resistant TB in Klang Valley is the East Asian Beijing clone. This is the lineage 2, which comprises of 71% of total sample. Next is the Indo-Oceanic, which is lineage 1, comprised of 18%. Euro-American, Lam T. Ural, lineage 4, which comprises of 9%. And East African Indian, clone Chas, lineage 3 of 2%. There was 29 isolates linked in 12 clusters identified, and 11 clusters were actually differed by 1 to 5 allele, which suggesting of recent transmission. And there was also revealed a new possible epid link, which was previously undetected by conventional investigation, which noted in 2 cases that were identified as Euro-American Lam clone that had patient were reside 12 kilometer radius from each other. However, no record of connection was made between the 2 patients. Findings number 3, there are drug-resistant mutation noted. The highest mutation was conferred to rifampicin resistant, which comprised of 16 cases, and the other cases were noted to have resistant were conferring to isoniazide and ethambutol. The most common resistant mutation was noted to be CATG 315 serine, which was noted to be 29 in frequency. The hotspot region of 81 base pair of rifampicin resistant determining region was also studied, and there were 7 loci noted to have single and multiple mutations on RPOB gene. And the most common mutation for rifampicin in RRDR region was noted to be serine 415 leucine, which comprised of 23 isolates, and these are associated with category 1, associated with resistant. And interestingly, there are 2 rare mutations outside rifampicin-resistant determining region, which are valine 470 phenylalanine, 3 isolate, and isoleucine 491 phenylalanine, that is 1 isolate. So these 2 mutations are not being covered by rapid molecular tool, as rapid molecular tool only covering the RRDR region. So unfortunately, these 2 mutations, valine 170 phenylalanine and isoleucine 491 phenylalanine, are both associated with resistant final confidence grading 1. And in 2009, Setini et al. Has reported that 30% of MDR-TB strain isolated in South Africa were found to be carrying the RPOB isoleucine 491 phenylalanine silent mutation. And this could lead to ineffective treatment and RRDR mutation detection. Next is the targeted generation sequencing on drug-resistant TB. So TNGS, or targeted next generation sequencing, is defined as massively parallel sequencing to detect drug-resistant TB drugs, starting from clinical sample and ending with diagnostic report that relate to detection of MTBC mutation, whether they are present or absent of drug resistant, based on interpretation of WHO standard catalogue. WHO has recommended the use of TNGS in bacteriology confirmed pulmonary TB on pulmonary sample for certain drugs that are suboptimal for phenotypic results. And WHO has evaluated and endorsed 3 commercial kits for TNGS, which are Diplex MIG TB test, MPOR TB test from Oxford Nanopore, and TB6 from Hanzhou Shengting. We have evaluated Diplex kit for TNGS in comparison to WGS for the drug-resistant TB detection. And in Diplex, there are 4 main components that were being tested, which are quality control, identification of mycobacterium species using target gene of HSP65, genotyping using Spoligo typing method, and also mutation in 19 target genes for 13 drugs, which is associated with WHO mutation catalog. And a comparison of Diplex MIG-TB versus WGS were performed on 32 clinical isolates profile that are recognized as pan-susceptible, mono or poly-resistant, multi-drug resistant, as well as pre-extensively drug-resistant isolates. And the performance of TNGS Diplex for drug-resistant TB were noted to have high inaccuracy between 94 to 100%, high sensitivity 80 to 100%, and specificity 92 to 100% in reference to resistant profile by WGS. Diplex correctly identified almost all mutations in all isolate that are significantly associated with resistance in WGS, and it do not identify mutations which do not confer significant resistance. Now we are in discussion and conclusion part. So studies in West Malaysia has persistently showed that Beijing clones was one of the main lineage in drug-resistant TB isolate in West Malaysia compared to East Malaysia Sabah, whereby the most important or main predominant lineage is Indo-Oceanic. TB genotypes pattern in West Malaysia resemble pattern in study by Sasmono et al. 2021 in West Java, while in Sabah the genotype pattern was parallel to Phelan et al. And Problit et al. In Philippines. Beijing clones was noted to be carrying multi-drug resistant mutation, particularly for rifampicin and isoniazide, and this was noted in 3 main studies in West Malaysia. And this actually concur to previous studies in Southeast Asia and globally that are mentioning Beijing clone as the most widespread resistant clone, and associated with independent risk factor for treatment failure and relapse. There is a high concern of misdetection of rifampicin-resistant TB by rapid molecular tool. And this has been evidenced by a case study report that was published in Botswana, which showed that they have missed a delayed in detection of pre-XDR variant by using expert rapid molecular tool, which missed detection rifampicin resistant, which was carried by a mutation clone of RPOB, isoleucine 491 phenylalanine. So this study underscores that genotypic drug susceptibility test is important to be used in complement to phenotypic DST to improve detection of drug-resistant TB. So the learning points that we got from all these studies is that geographical factors, population density and urbanization play a crucial role in shaping the transmission dynamics of TB and its lineage, evident by the different genotype pattern in West Malaysia and East Malaysia. WGS is a powerful tool for surveillance as SNP and early information of MTB complex may enhance delineation of transmission and improve discriminative limitation of conventional genotyping methods which are used now, which is miru-VNTR and oligotyping. TNGS provide rapid and comprehensive genomic DST to complement phenotypic DST results for better accuracy in management of drug-resistant TB. What are the challenges to perform NGS tests? First is that we need to improve and enhance the resources, particularly budget allocation for reagents and equipments, skilled personnel and training. Second is the needs for strengthening the strategies to enhance coordination and work plan arrangement. These include logistical challenge, sample transportation, standardization of protocols, and ensuring quality control. Future direction. First, it is a need to integrate WGS into the National TB Surveillance Program through regional and phased approach, prioritizing the region with high TB burden as well as case clusters that involving drug-resistant TB. Second, to initiate operational research on TNGS for diagnostic by TB Lab IMR in 2025 to establish workflow and optimize the operational framework and SOPs. Third, the need for a paradigm shift among stakeholders to view the use of NGS in TB as an investment rather than expenditure. Okay. Last but not least, the conclusion is next generation sequencing NGS is a pivotal advancement in the management of drug-resistant TB and TB surveillance, providing a transformative approach for improved diagnosis and control in Malaysian current TB landscape. Thank you for your kind attention. If you have any suggestion or question, you can email to this email address. Thank you.
Thank you, Dr. Siti, for the insightful presentation. We will now be moving to questions and answer session. Kindly send your questions using the link provided in the YouTube channel, and we will try our best to connect you to our distinguished speakers through the posted questions. Nevertheless, should the time does not allow us to address all the questions, our speakers will try their best to answer the questions through email. So I think we have received a few questions from our audience. So I think the first question to Dr. Azura, what is the available test to diagnose TB infection among children, and what are the challenges?
Right, very good question. Thank you, Dr. Nani. And thank you for the person who asked this question. Right, to diagnose TB among children patients, it's quite challenging actually. But for the diagnosis, of course, we have to follow the standard. Number one is AFB smear. and then followed by culture, and then gene expert. Now we call it ultra, gene expert ultra. And then the challenges actually for children is having good samples or good specimens, because we know that children very difficult to produce sputum. So what we can do is instead of using sputum, we can use other specimens such as gastric lavage specimen. And then, as we know that specimen from children is paucibacillary specimen, which is number of bacilli is very, very little. So that now the best diagnosis is expert, gene expert ultra.
Okay, thank you, Dr. Azura.
You're most welcome.
Now we have also a question to Dr. Siti. How has genomic sequencing helped in understanding the mechanisms behind drug-resistant TB in Malaysia? And what are the key findings that could inform more targeted treatment strategies in the region? Over to you, Dr. Siti. Thank you.
Thank you very much for the questions. So the question was asking on the genomic sequencing role in understanding the mechanism behind drug resistance, and what are the contribution that TNGS can be, what are the contribution of TNGS in making a successful TB management. Okay, so as we know, genomic sequencing using NGS is a very advanced technology nowadays, and from the TNGS we may get the comprehensive drug-resistant mutations results compared to rapid molecular tool or a phenotypic drug-resistant DST. So by using TNGS results, we may target the accurate, we may plan for accurate and comprehensive management in drug-resistant TB cases by getting the right treatment at the right time. So for example, we can get the whole results of drug-resistant anti-TB by having just one test, and from there the clinician may predict what drug that can be used and which drug that cannot be used in that particular patient from the first setting. Thank you.
Thank you, Dr. Siti. Maybe we move on to the next question to Dr. Azura. So the question is, how does the cost of point-of-care testing, POCT, compare to lab IGRA, considering the cost is a key concern with lab IGRA at the moment? Over to you, Dr. Azura.
Thank you. Thank you, Eslio. Very tricky question, actually. Right, when we are talking about cost between POCT compared to lab IGRA, okay, when we are talking POCT, it won't involve any equipment. It's just like, rapid test kit. But when we are talking about IGRA test, we need equipment. It's quite expensive. If, let's say, inshallah, in future, our POCT successfully developed, it will be much, much cheaper compared to the IGRA, inshallah.
Okay. Thank you, Dr. Azura. Okay, I think we still have time for another question to Dr. Siti. How can targeted next-gene sequencing, or TNGS, improve the accuracy and turnaround time of that susceptibility testing, or DST, for TB in a high-burden setting? Over to you, Dr. Siti.
Thank you for the second question. So for the accuracy part, I think I've elaborated in the previous question. So for the shortened turnaround time of DST, as we know, TNGS is using clinical samples compared to phenotypic drug susceptibility test, which needs the growth of MTB culture using liquid or the solid media. So from here, we can already guess that the TAT for TNGS is much, much earlier that can be established from the clinical samples.
Thank you, Dr. Siti. Yeah, another question to Dr. Azura. In your opinion, why are we having MDR-TB on the rise? Thank you.
Right. Very, very good questions. Right. Thank you, Sakina from KKM. Okay, based on my opinion, why we do have MDR-TB on rise? Okay, number one, all MTB drug take long-term regime. It will take up to 6 months. So I don't think that most of the people will finish that course. That is number one. So we know that if we skip our antibiotic, it can cause MDR-TB, it can cause resistance. Number two, probably based on my opinion, I might postulate this answer actually. The MDR-TB rise because cases rise, maybe probably because of the, you know, the effectiveness of the drug itself in penetrating Mycobacterium cell wall. Because you know, Mycobacterium cell wall from time to time can mutate to adapt with a different environment. Probably it contribute to that kind of thing. That is my postulation. Thank you.
Thank you, Dr. Azura, for your answers. We still have time for another question to Dr. Siti. And this time is from Ms. Hani Emma Fasni, IUM. The question is, how can genetic information contribute to disease tracking in Malaysia and help reduce the incidence of antimicrobial resistance in TB? Over to you, Dr. Siti.
Thank you very much for this interesting question. So genetic information is very useful in tracking disease, especially the close contact of TB patients. As I mentioned earlier, the SNPs and allele information that we can get through WGS is a very, very powerful tool in determining the transmissibility study between similar clone TB strains. So in this case, we can have a better high resolution genotyping and also epidemiological study. And for antimicrobial resistance, as I mentioned earlier, if we can get a comprehensive drug resistant pattern or results at the first place, we can avoid the use of unnecessary TB drugs at the first time. Thank you.
Thank you, Dr. Siti. We have another question to Dr. Azura. In addition to targeting peptidoglycan, could targeting tryptophan synthesis also kill MTB?
Right, thank you. From Dr. Aswir IMR. Thank you for the questions. I think to answer these questions, maybe we have to do research together later. But you know, maybe I can give some idea lah to answer these questions. Instead of targeting peptidoglycan, we can target other amino acids as well, if that amino acid is very important for its metabolic pathway. For example, histidine. But tryptophan, I'm not very sure about that. But probably we can discuss further to do research together. Thank you.
Okay, thank you, Dr. Azura. I have another question. This probably can be answered either by Dr. Azura or Dr. Siti, and this is from Tilak, TNDI. So TB is high prevalence among marginalized communities. How do you engage with them to get more samples? It's important to include them because they are part of our society. Thank you.
Maybe I can answer this first. Okay, so TB is indeed a high prevalence among the marginalized communities, and it is very important for us to get them for detection of TB, and this can be done by continuous education as well as public health engagement. So more TB programs should be done, should be started in our community level, so that marginalized communities especially can get the real information, as well as being checked for their status for further intervention. Thank you.
Thank you, Dr. Siti. Dr. Azura, would you like to add anything to this question?
Okay, I think yes, it's good to include them to get more samples, but I think we have to also discuss with bahagian kawalan penyakit for and policymakers to get it done. Thank you.
Okay, it seems that we still have a time for another question. Maybe I can ask this to Dr. Azura. In terms of detecting latent TB, how good is IGRA now? Is it widely used to any patients in Malaysia? Thank you.
Right, for IGRA, actually it's quite good, but the limit of IGRA is the predictive value of it, because it cannot differentiate between active and latent. That is the main problem. But so far, IGRA, if we don't have any option, so we should proceed instead of using man-to test, because man-to test has much lower sensitivity and specificity compared to IGRA.
Thank you, Dr. Azura. Another question to Dr. Siti. Do you think it's necessary to have a screening of TB among healthcare workers in here in Malaysia? Thank you.
Screening of TB among healthcare workers.
Yes.
So this is a very, very important topic, whereby we actually have a guideline for this. So healthcare workers are the main target group that we need to screen for latent TB as well as active TB, because they are engaging closely with patients, and this made them very vulnerable to TB. And we have started healthcare worker screening in NIH itself, and we supported the use of IGRA in screening healthcare worker TB among healthcare workers.
Okay, thank you, Dr. Siti. And then we have another question from Aslin, ICR, to Dr. Azura. Is point-of-care testing feasible for the implementation at the primary care settings?
Right, thank you, Aslin. Again, yeah, I think we can be a good collaborator in future, inshallah. So is POCT feasible for implementation in primary care settings? Yes, of course, because it's just like, very rapid test kit. It's very cheap. Don't need expensive equipment, don't need very big space, and we don't have a very trained or skilled person to perform that test.
Okay, thank you, Dr. Azura, for the answer. I just would like to ask both speakers, is there anything that we would like to add to our discussion today for our final words for this webinar?
So,
Dr. Azura, yeah.
Right. So since that in IMR we are doing a lot of research regarding Mycobacterium tuberculosis from basic to clinical. So I would like to invite any researchers, clinicians, from stakeholders, from hospitals, from university, come to IMR, NIH, and we can sit to discuss further about collaboration, and we can fill up the gap in research to make sure that we can contribute significant contribution to our country to improve the health system in Malaysia, especially in combating tuberculosis infection. Thank you.
Thank you. And how about you, Dr. Siti?
Okay, resonating on the theme for our webinar today, that is translating the research to practice. So I would like to inform that we in TB Lab IMR, we're going to start the operational research on TNGS by second half of this year. And we hope that by initiating the operational research in TNGS that we can contribute to better diagnosis of drug-resistant TB, which we can have both genotypic and phenotypic results, DST results for the appropriate patients. Thank you.
Thank you, Dr. Siti. I'm afraid that this is all that we have for the question and answer session. Any additional questions will be answered by speakers through email. And we hope that this webinar has shed light on some research work that we are conducting here in IMR and would spark interest and co-initiate research collaborations with others. Ladies and gentlemen, now we have come to the end of the webinar session. I would like to thank all participants for their support to make this session successful. Don't forget to subscribe to our National Institutes of Health Malaysia YouTube channel. Before I conclude, I would like to announce that we will have the next session of IMR-SEAMEO Webinar Series on 28 February 2025, so mark your calendars. The theme for the next webinar is the 3 tiny blood-sucking insect, a Mohd Noor. We will also have a phage therapy symposium, which will be conducted online on the 12th February 2025, the morning session. Do share your feedback on this webinar series by scanning this QR code. We hope you can join us at the next sessions. Until then, many thanks from all of us at IMR. Take care and stay safe.